So we often talk about control and elimination as if these are part of the same continuum, only differing in the scale of the outcome that's achieved. But these are very different processes, they have different priorities, and as such, they have very different requirements in order to achieve those priorities. So they take a completely different approach. In thinking about control, the goal is to scale up interventions, to increase access, increase efficacy. We're probably measuring transmission and wanting to make sure that we're having an impact on reducing transmission. And oftentimes, control programs are run by, on disease-specific silos. So this is just a generalization, but typically these are run by one disease control program. As we move to pre-elimination and we've reduced prevalence, what we've ended up with is possibly increased heterogeneity. So we need to identify those clusters. They may be geographic clusters or they may be social clusters. The role of asymptomatic individuals and individuals with low density infections becomes even more important. So it's about having the tools to sensitively detect those individuals and having the knowledge that we can safely scale down interventions in areas where we've achieved that transmission breakpoint. And then thinking about elimination, we need to then have the tools to validate the absence of disease. And this is much harder than it sounds. I mean, how many negative samples do you need to collect before you're confident that the true answer is 0? It's about being prepared to detect resurgence and having a rapid response. So some of the challenges with surveillance of vector-borne diseases. Well, with neglected tropical diseases, many of these don't elicit treatment seeking right after infection. There may be a lot of asymptomatic individuals, or there may be a very long period from the time the infection is picked up to when there's morbidity associated with that infection. All the while, transmission has been going in that community. And as I said, by definition, as you reduce your prevalence, it becomes very difficult to detect any remaining cases, and the cost per case detected rises significantly. So communities and donors may lose a bit of momentum here when you have a disease that's of very, very low prevalence. We also see correlations between individuals that are highly exposed, also having very poor access, and also poor participation in surveillance activities. So that when the team turns up to a community to take a sample and measure the microfilaria prevalence in the case of filariasis, you may not be seeing the individuals that hold the greatest burden of disease. And in the long term, specifically screening for something that isn't there but that may reemerge or come across the border, it's very costly and it will be low on the priority list for many ministries of health. But this is necessary in order to preserve the gains that have been made by control programs. So giving some examples from filariasis, which is the main disease that I work with, the goal is to eliminate LF by 2020 as a public health problem. And this is done by mass drug administration, at least 5 years with at least 65% coverage. And the MDA targets the juvenile stages, so it must be maintained for the duration of the adult worm's lifespan. The scale-up of MDA has been tremendously successful. Billions of drugs have been donated and distributed, and this has prevented approximately 97 million cases so far. And here I've showed the timeline. Once you've had a number of rounds of MDA, and pre-task tells you that the microfilaria prevalence is below 1%, then there's a switch to the transmission assessment survey. And this is kind of the turning point between MDA and post-MDA surveillance. So TAS, these involves screening school children for the presence of the adult worm antigen, which would indicate that they've been exposed in their lifetime since that control program started. So the transmission assessment survey is one of the best tools that we have for confirming filariasis elimination, but it's not going to be the only tool, and we really need additional tools in order to complement the tasks. So I think this map really nicely highlights the successes so far as well as some of the challenges. So all of these blue dots are areas where current LF prevalence is 0. And the yellow dots are areas that are coming up right behind with relatively low prevalence, nearing that 1% threshold to move to the transmission assessment survey. But yeah, I'll highlight the example of Togo, which has a lot of blue dots, but is surrounded by areas that are still needing to scale up MDA and where there still is active transmission. So for these blue dots, it's going to be very important to ensure that transmission remains very, very low while neighboring countries are working on their elimination program. And with all of these yellow dots, it's about sensitively measuring that 1% prevalence and moving to the transmission assessment surveys as well as the post-MDA surveillance. So I'm a vector biologist, and my contribution to the multidisciplinarity is how can we use mosquitoes to give us more information about elimination programs. So xenomonitoring literally means diagnosis via a stranger. And this is an indirect assessment of pathogen prevalence in a community. This does not tell us about transmission, but it does give us a real-time indication of parasite presence compared to, say, for filariasis antigen testing, which tells us about previous exposure. And I think one benefit here is that mosquitoes are able to reach the highly exposed individuals, even those that are not accessing treatments and that are not turning up for surveillance activities. In fact, they may even be more biased to feed on individuals that are highly exposed. And DNA is present in vectors as well as non-vectors. For some vector-borne diseases, it's very difficult to collect vectors. I'll say HAT is a good example. But mosquitoes, if they're feeding on an individual that has that infection, they're going to pick up DNA as well. So I just wanted to give an example of how I've used xenomonitoring data in some of my own work. So this is from Papua New Guinea, where we were looking at transmission before and after LLIN distribution. And what I've shown here, this is not xenomonitoring, but this is transmission monitoring, where we've dissected mosquitoes and we've quantified the number of developing larvae or infective stage larvae that are found. And you can see in a very highly endemic area, prevalence of a few percent in mosquitoes, and the hashed area is the individuals that had infective larvae, so around 0.5%. And then after the nets went out, we found in 3 of these villages, we didn't find any mosquitoes with developing larvae, and we found very few mosquitoes in those other 2 villages. None of them were infective. So this tells us that currently there is very little, there's undetectable transmission. But we also took half of our samples and we extracted DNA and we looked for the DNA for Wuchereria bancrofti. And you can see there's a 10-fold increase in the prevalence of DNA compared to what we saw in the developing larvae. Now, after bed nets, we don't really see a difference here, and we don't expect to see a difference because this is not an intervention that targeted microfilaria in the community. So what this is telling us is there are still a lot of individuals in this community that have microfilaria. Of course, what we want to be able to do is correlate that with what's the actual prevalence in humans. Sorry. Oh. Okay, here we go. Yeah, thanks. Okay, so what I've done is I've taken this data, the infection rate in mosquitoes and the MF prevalence in humans, and I've also added from a few other studies that did this as well. Now, these are taken from different locations, different vector species, and different methods of collection. But there's not a lot of data out there presenting both MF prevalence in humans and DNA prevalence in mosquitoes, because in the control stages of a program, we want to measure transmission, not just presence of DNA. And I've split this into 2 because on the right-hand side, that's a study that was using a very different method, a biased method of only screening mosquitoes that were blood-fed. So there's a correlation here, but there's quite a bit of movement around that line. And that variation, again, is due to different locations and species and collection methods. And I will come back to that. So I think xenomonitoring is a great tool that could be used to kind of supplement decision-making around the end game for control programs, but there's a number of reasons why it's not used at scale. Frankly, a lot of programs that just have other priorities, and bothering with collecting mosquitoes is a bit too much. Identifying mosquitoes requires time and expertise. Processing those mosquitoes also requires time, and if you're doing molecular detection, additional equipment is required. I think one other important limitation is that we don't have standard guidelines about how many mosquitoes do we need to screen and how should we collect them and what should that method be. And we don't really have a way of correlating that result with something that's meaningful for human disease prevalence in humans. So I'm going to talk about a few new developments in the field of xenomonitoring, starting with a project that we're doing, vector excreta surveillance, and you may have seen this from Darren's poster. But this study aims to reduce the time, costs, and the expertise needed to screen mosquitoes for pathogen DNA or RNA. And we'd like to determine whether mosquitoes could be used for xenomonitoring for non-mosquito-borne pathogens. And we're taking advantage of the behavior that mosquitoes have of going out there, taking a blood meal, and delivering a nice, clean, concentrated drop of feces or excreta, which we can then screen. So in this picture, you can see that that mosquito, as she's taking a blood meal, she's concentrating that. And what's coming out, this actually is very unlikely to be positive for any pathogen DNA. But over the next at least 5 days or so, she's going to be excreting tiny droplets of that digested blood as well as excreta. So just to give some highlights of work from my group as well as from colleagues, in my group we've been exposing mosquitoes to infected blood meals. We've been doing it with Brugia malayi, Trypanosome brucei, which of course is not mosquito-borne, as well as Plasmodium falciparum. And what we find is that the DNA is detected in that excreta for a number of days after relatively low density infections. And colleagues that were working kind of in larger cages, they found that you could have a pool size of at least 500 of mosquitoes that were exposed to uninfected blood with one that was exposed to infected blood. So the pool size here is significantly higher than what could be achieved with grinding up mosquitoes. And researchers at Institut Pasteur working on dengue also find that dengue can be picked up in the excreta for quite a while after exposure. So this is all quite interesting, but the methods used here are quite cumbersome. So on the right, they fed mosquitoes blue sugar so that they could then punch out the excreta drops. That's not really feasible. And then in some of our work, when we're trying to get them to defecate into a tube so we could take that straight to PCR, that's also not scalable. And so what Darren had presented is an adaptation to mosquito collection cups, which we routinely use. It's a paper cone that can be cut to size and really put in any kind of collection cup. And it's sprayed with a commercial product which gives it a super hydrophobic coating. And any of those tiny droplets all end up at the bottom of that cone, which you can have go straight into your tube for PCR or straight into FTA card if you wanted to preserve it for longer term. And we found that this works very well, and we can pick up DNA from singly exposed mosquitoes at relatively low densities, and these cones can be reused. There's no contamination. So we're excited about this, but of course a number of other things need to happen in order for us to do this, and we'd like mosquitoes to come to us. So I'll mention a few developments. This is not my own work, but passive trap design. There are many ideas for new mosquito traps depending on what the purpose is, and this is one that I just saw recently. Researchers at James Cook University have modified the CDC light trap, a very low-tech adaptation, but that would allow them to kind of swab this container. On the right, it's a very high-tech example. This is probably an overly complicated solution to a not very complicated problem, but Project Premonition is using drones to deliver these sort of mosquito hotels. Mosquitoes are attracted to this, and they have lasers that can determine the sex and the species of that mosquito, and they can trap them in there, and the data goes straight to a cloud somewhere. Again, I think maybe somewhere in between here, we do need further development and thinking about how we can get the most information from vectors out there. And then this is a Gates-funded project with colleagues at Smith College, Steve Williams and Nils Pilote, where we're trying to make this approach even more field-friendly. So they've developed DNA extraction techniques which can be done without a centrifuge. In this example, they're grinding up 25 mosquitoes, 20 minutes total procedure. With the excreta surveillance, of course, the step would be even easier, and we're getting some success with direct PCR, so that would skip the extractions altogether. They're also looking at using the mini PCR, which gets the job done in about 60 minutes, and it runs off of a small solar cell and a cell phone. And all of these things can easily fit together in a backpack if you wanted to do this sort of on the spot. And then in terms of the detection, they've been developing a test strip detection for Brugia malayi, which we'll then be using for Wuchereria bancrofti. And this has a very quick incubation step, and it works just like pregnancy tests, antigen tests, and things that we often use, where you have a control line and a positive line. And this is also a commercial product. So we think that these developments are bringing xenomonitoring in closer reach. But again, the question of what do these data mean So we've been working with Deirdre Hollingsworth and Emma Davis at University of Warwick. So Emma's been doing some nice modeling of mosquito life history traits, and we're using those models to then look at the encounter rate in a community with infected individuals to get an idea of what proportion of that mosquito population is likely to be positive. And this is going to be dependent on a number of different things, but probably one of the most important things is the parity rate in the mosquitoes that are collected. So if you're collecting young mosquitoes, then they're much less likely to be positive for that pathogen compared to older mosquitoes. And I think a lot of the variation that we see from village to village and between different collection methods is down to the fact that we're biasing our collections towards older mosquitoes or younger mosquitoes. But I think what we're really interested in is what's happening down at the low host prevalence. So just as an example here, this shows you some predictions about the vector prevalence at low host prevalence, and you can see that this is definitely quite feasible. Around 5% prevalence or less, we're looking at 5 to 10% prevalence in mosquitoes if we're using a tool that is biased towards older mosquitoes, and we do have tools like that, such as gravid traps. So we'll be working on these estimates more. And we're also going to be validating these within an endemic setting in Ghana, where we have relatively low prevalence of filariasis. So I think I've demonstrated the potential for a number of diseases that are nearing elimination. But I wonder if this could also serve a purpose for early detection for outbreaks. So this is just an example of chikungunya outbreaks around the world. And this graph shows you Zika, and the dark red areas show you the areas where there's a higher risk of Zika transmission. And the size of these circles shows you the number of people that are coming into these ports from areas with Zika transmission. And we already have quite a bit of surveillance around ports, and they collect many, many mosquitoes, and they send them out for processing different purposes. And so we wonder if this is something that could also be layered on there to screen for various viruses or other pathogens that could be imported. So to conclude, we need an early alert tool to detect emergent and resurgent vector-borne diseases. We will require cross-disease collaborations in order to take us to the end game. So thinking about a tool that allows us to screen for multiple pathogens at the same time. We think that excreta surveillance is a low-cost, non-invasive, and adaptable approach that does have some utility, especially for these very, very low prevalent infections that we want to confirm an absence. And with the potential to detect any bloodborne pathogen, we really want to explore what else we can do for this. And so in addition to non-mosquito-borne diseases, what about non-vector-borne diseases? And we're also looking to see whether we can use this to screen for certain alleles, such as drug-resistant alleles, in a community to get more information about the parasite population. And I realized I've taken 20 minutes of your time without a single parasite photo, so I've just put this in at the last minute. That's a filarial worm coming out of a mosquito's proboscis. And I'd like to acknowledge funding from the MRC, and I'm also supported by the NTD Modeling Consortium and the Countdown Consortium. Thanks.